Hello, my name is Constantinos Mikelis. I'm an Adjunct Associate Professor at the School of Pharmacy at Texas Tech University Health Sciences Center.
And I'm Ulrich Bickel. I'm professor in the Department of Pharmaceutical Sciences at the School of Pharmacy here in Amarillo and Associate Dean for Sciences. Well, the type of research is, as the name implies, mostly pharmaceutical sciences. So that is a wide field. So we have colleagues working in drug development, medicinal chemistry, finding new drugs, and then testing toxicity, for example, or effect of drugs in the cell culture. We were looking for instruments that can do live cell imaging in a way that you can monitor cells growing in a culture dish for long periods of time without changing the cell metabolism or damaging them. This is a new technology that uses very, very low intensity level light, no fluorescent dyes, which are also potentially toxic. And that is basically, at this point, the only instrument that has these qualifications. So it was a natural choice.
I have not seen another system that is able to do the same things in the same manner.
Another part of our faculty is centered on cancer research, and that is actually where this instrument was funded by the Center for Cancer Research. Growth phases in cancer research, wound assays, wound healing assays, that's a very common experimental setup. And also we are trying to use it for our blood-brain barrier research, and that is something in development, monolayers of endothelial cells, for example, and how they grow. And we have a core facility here, and that's part of this core facility for imaging purposes.
In our group, we combine both vascular and cancer research, so we use it for angiogenesis development, and we use it also for cell communication purposes. It is very difficult to be able to quantify and to do single cell analysis by hand. You will get a number of cells, but you will not be able to capture all these cells that they are captured with Livecyte. So it allows us to generate a significant amount of data in limited time.
The advantage is really that you have huge numbers, so you have very good statistics. So you can detect effects that are objectively verified. So this is basically building the system that you have thousands, tens of thousands of cells at same or different conditions at the same time, which is not possible to do in a manual. And because it's time-saving compared to visual and manual analysis, it is also then easier to accommodate a number of different labs.
In terms of experiments, it can cover a wide variety of topics, which is advantageous also to incorporate many different groups to use it. So this basically makes it one ideal instrument that a core facility can have. The vast majority of our work, it is involved. So at the end of the day, I think it will contribute to several publications of our group and of several other groups from what I know. I think that the value that this machine provides in terms of research is very significant. We were amazed that when we received the microscope, that was during the pandemic, and even though the company is UK based, they were able to send everything to make things easy for us to assemble it. They are very helpful, and they are basically constantly next to our site. They constantly developed. So they have created new developments for angiogenesis, which was the focus of my group. So they have updated the software. It shows that they are continuously trying to advance the system. I have not seen another system that is able to do the same things in the same manner.